Genetics Matters, Episode 16. My hypothesis was wrong. Fantastic. Welcome back. I'm Brad Goodner. As we go through the key ideas in genetics that will help us solve the problems of the future, we focus on the experiments of a few people who have shaped our thinking in a major way. So far, those folks have included Frederick Griffith, Oswald Avery, Erwin Chargaff, Jim Watson and Francis Crick, Charles Janosky, and George Beadle and Earl Tatum. Now let us turn to Jacques Monod. He may not be familiar to most of you, but if you want, to paraphrase his words, to understand how an elephant develops, then you better know Monod's work on E. coli. Born, raised, and educated in France, Monod's early career seems rather rootless. He spent some time at Caltech studying Drosophila genetics, but in reality, spending his creative energies conducting a local chamber orchestra. He was an outstanding cellist. Returning home to Paris, he worked on growth and nutrition of protozoa and finally switched his attention to bacteria. During World War II, his experiments often had to take a backseat to his work as a member of the French underground. Later in his life, Monod played a major role in the social issues of his time. A truly interesting fellow. Monod's work that concerns us here was to understand the role that sugars play in determining the growth rate of the bacterium E. coli. If this does not sound like the secret of life to you, it probably seemed initially not much more interesting to Monod. It is what he did with the subject that commands our attention. His initial experiments gave no surprises. Give food to bacteria and they grew pretty much as you might expect. They grew with an S-shaped growth curve until they exhausted the sugar, and then they stopped. The maximum exponential growth rate depended on the sugar added. Some sugars, like glucose, supported the maximal rate of growth, while other sugars were not as effective. Once he had gone through all the obvious sugars, what could he do for an encore? Well, there are the combinations. What's the rate of growth if you mix two different sugars and so forth? Gripping stuff, don't you think? Actually, the story became much more interesting than you might think when Monod grew E. coli on a 1-to-1 mixture of the sugars glucose and lactose. Monod observed a double S-shaped growth curve. This type of growth is called dioxic for its two growth periods. Oxy means to grow. What experiments could you do to get a handle on what's going on? Well, you could vary the ratio of glucose to lactose. When Monod did that, it appeared that the bacteria was eating its preferred food, glucose, first. There's a pause, and then it would eat all the lactose. At the time of Monod's experiments, there was intense interest in biochemistry, especially on how protein enzymes interact with their substrates. Given this interest, it is not surprising that Monod's initial explanation for dioxic growth was in terms of enzymatic activity. Indeed, when the ability to break down lactose was measured, in E. coli, the enzyme involved is called beta-galactosidase. It was found only during the second stage of growth. It must be noted that enzyme activity is the capacity for the enzymatic reaction to take place, whether or not that capacity is actually utilized. Imagine a factory that may or may not be used, even though all the machinery is there. Monod spent much of the rest of his scientific life explaining how the presence of the two sugars, glucose and lactose, influenced beta-galactosidase activity. It became evident that there were two effects. 1, lactose is required for the appearance of beta-galactosidase activity, and 2, glucose prevents the appearance of beta-galactosidase activity despite the presence of lactose. To explain these effects, Monod initially put forth an explanation based on current thinking about protein binding to substrates. It was thought that enzymes got their remarkable specificities by wrapping themselves around the molecules they bound to, like making a mold of a key. Monod postulated that there exists a universal pre-enzyme that wraps itself around glucose when glucose is present, and in that form is able to break down glucose. When glucose is consumed, the enzyme reforms itself by wrapping around lactose and adapts its activity so it can break down lactose, but not glucose. It is important to see that there are two functions envisioned for lactose in this model. One, lactose as an inducer of a new enzyme shape, and two, lactose as a substrate for the enzymatic reaction. In Monod's model, the two functions were achieved in the same way, by lactose interacting with a single protein, beta-galactosidase. It turns out Monod's view was wrong, but it was his own research group that proved that they were wrong. We now know, of course, that the function of a particular enzyme is determined by the amino acid sequence encoded by its particular gene. Proteins are not generic putty that learn their shapes and function by wrapping around the chemicals they act on. Nonetheless, small molecules like lactose exert enormous influence on activities within a cell, and they do so by binding to protein. How do we get at the role of lactose as both an inducer of beta-galactosidase activity and as a substrate for the enzyme? The answer came in the form of chemical derivatives of lactose. To see the basis for this insight, it is necessary to learn a bit about lactose. Lactose is a disaccharide composed of two sugar monomers. Beta-galactosidase splits lactose into its component sugars, glucose and galactose. The enzyme is called beta-galactosidase because it acts on beta-galactosides, compounds in which something is attached to galactose by a beta linkage. Monod's original idea about a malleable enzyme suggested that any molecule that was a substrate of beta-galactosidase would also be an inducer, and vice versa, and that both of these characteristics involved the compound binding to the enzyme. Monod sought to test this hypothesis by sending out graduate students and postdoctoral fellows to organic chemistry labs across Europe to construct lactose derivatives. They sent back compounds for separate tests for possible inducing or substrate activity. The impact of those results on Monod's thinking were striking, as shown best in a short telegram that Monod sent on October 14, 1950, to Melvin Cohn, who had been sent to a lab in England. The message concerns the compound phenyl beta-thiogalactoside, or PTG. My Texan version of French is terrible, so I will translate the telegram. Affinity, very high, meaning that PTG showed strong binding to the enzyme. No hydrolysis, meaning PTG was not a substrate for beta-galactosidase. No induction, meaning PTG could not induce a change in enzyme shape. Fantastic. Signed, Jacques. Using many different lactose derivatives, Monod and his colleagues figured out that there were compounds that were both inducers and substrates, such as lactose, compounds that can induce but never be substrates, such as IPTG, compounds that could not induce but could be substrates, such as ONPG and PGAL, and some compounds that could bind the enzyme but could never act as either an inducer or a substrate, such as the PTG that was mentioned in the telegram. Remember that Monod's hypothesis was that all inducers would be substrates and that all substrates would be inducers. The data said overwhelmingly that he was wrong, and what was his response? Fantastic. I am wrong, but that is fantastic. Can you see why Monod thought that these results were fantastic? Clearly, the two functions of lactose, inducer and substrate, are clearly different, and it was much more likely that there were two different proteins binding lactose, one involved in induction, whatever that means now, and the other one being the enzyme beta-lactidase. These results and Monod's responses to them set the stage for the discovery of gene regulation. More on that next time on Genetics Matters.